Hey there, everybody. I'm quickly going to go over this is going to be a short video talking about colony morphology, just so you are prepared for lab. And I'm using this website. This is not my website, but they have colony morphology explained relatively well. So what you're going to be doing in lab is you're going to be looking at bacteria on a Petri plate, and you're going to use a technique known as colony morphology to determine certain characteristics that differ between different groups of bacteria. So you're going to have, I think we have a total of 19 species of bacteria in our lab, but one of the species I don't have growing right now because I just can't get it to grow. So I think you'll have like 18 different Petri plates that you'll be looking at. And you're going to look at characteristics of those plates. Some of those characteristics are going to be if the edges are circular, or if the colonies look circular, if they are irregular in shape, if they have a filamentous structure, do they kind of look rhizoid or fungal-like? Are the colonies elevated in any way? So if they're not, they're flat. If they have like a little bump, they're umbonate. What does the edge of the colony looks like? So does the edge look very bumpy? Is it smooth? Are there curled regions of the edge? And so the thing about this is your mechanism. This is going to be a paper that you're going to do. I will grade it, but you're going to write this stuff down for you because this is what you're going to use to help you identify your unknowns. So if we look at this plate here, I'm not sure if I can make it bigger. I can't. Okay, I was going to try, but I don't know how to use computers good enough to make things get really big or small. Some of the other characteristics you're going to look at are the size. So are they large? Are they small? What is the color? So some colonies are going to be very white. Some are going to be more opaque. Some might be yellow or red or purple. It just depends on the species. And then texture. You're not going to physically touch them, but I want you to look at them and determine if they have a specific type of texture. Do they look dry and rough? Do they look really mucousy? So if you look at this plate here, this is somebody coughed on a Petri plate, and you can see on this one you have these very bumpy edges. And I thought I saw one that had the curled edges, but maybe I didn't. You see a bump here, an umbonate, where there's a bump in the middle. You have yellow, you have very tiny, you have very large colonies, you have white colonies. So there's lots of different types of colonies on here that you can see. And you can look at other examples as well. So I've made the plates last week, so you'll have fresh bacteria to look at. You're going to use magnifying glasses, and you're going to look at the bacteria on the plate and write some very descriptive terms. Draw a picture of the bacteria so you know what they look like, and then you're going to keep that for when you're doing your unknowns. Okay? Talk to you later. Bye.